All right, welcome back. In this video, we're going to go over protein folding and structures. I am going to blow your mind in this video, well, hopefully. But in order for me to blow your mind, we have to get there first. But before we get there first, I want to be that guy. I know. But hold on, let's break this down for a second. Not subscribed, 72.9%. Subscribed, 27%. Help a brother out, come on now. I'm kidding. Thank you so much for the support so far. Let's see that. We have 3 months in, 12,000 views, and 138 subscribers, or 137 subscribers. That's amazing. Thank you so much. I love doing this. And if this helps you guys, I'm happy. But let's bump this up, a little bit. All right, let's do this. In order for amino acids to turn into proteins, they must fold. A lot. Well, there are 4 different levels in protein folding. The first, or level 1, is the primary structure. The primary structure of proteins is the amino acid chain. Level 2 is the secondary structure, called Alpha Helices and Beta Sheets. And all of these levels we're going to go over in depth, step by step. So don't worry about it. This is just an overview. Level 3 is the tertiary structure. This is when the amino acids fold into a 3D shape and make a subunit. The quaternary structure, or level 4, is when multiple subunits come together to make a protein. So what I drew here on the right-hand side is what a protein actually looks like. It's probably something you didn't expect. It looks like a bunch of spirally noodles. That's literally what protein looks like. It's pretty crazy. So let's do the level 1, primary structure. The string of amino acids is the primary structure, also known as the polypeptide chain. I made a video not too long ago on the condensation reaction between amino acids, when amino acids join together. When amino acids join together, they form something called a peptide bond and release water. So imagine this reaction, but hundreds of times, thousands of times. We're joining hundreds and thousands of amino acids together to create a huge chain. That's what we're doing. And here we go. The result is a huge chain. Every single circle in different colors is a different amino acid. We formed a huge polypeptide chain. If you zoom in here, like one of the circles could be histidine, another one could be aspartate, for example. It could be glycine, it could be whatever. And the chain is random. It's whatever our body needs, whatever our body creates. This chain is completely random. And that's level 1. So let's do level 2. This is the new stuff now. So the polypeptide chain we just created can be folded into 2 ways. One is called alpha helices. Alpha helices are basically a spiral. So due to hydrogen bonds forming between amino acids in that polypeptide chain, it can literally fold into a spiral-looking shape. The other shape it could take is beta sheets or Beta-Pleaded Sheets. Same thing. Think about it when you fold a piece of paper and you fold it again and again and again. That's literally what beta sheets are. It folds into a flat-like structure, like paper. So on the left hand here, we have a alpha helice, and on the right hand side, we have a beta-pleated sheet. So what's happening here is maybe this section here turns into an alpha helix. Maybe this section here turns into a beta sheet. Then we got some remaining amino acids here that don't do anything. Then we got alpha helices here, then beta sheets here. Completely random. It's however the amino acids like to react with each other. So what I'm saying is that not this entire thing turns into an alpha helice or a beta sheet. Some parts of it do. The tertiary structure, or level 3, is when far away amino acids in that alpha helice or beta sheet we just created bond together. So they fold in and bond together. So it's like a 3D puzzle if you've ever done one. So it's a huge mess here. But let's break down what's happening. So we got a few alpha helices that have been created. One here, and then some here. We also got some beta sheets that have been produced. Here, here, and here. The yellow segments are the polypeptide chains, the stuff that actually did not become into an alpha helice or beta sheet. There's nothing wrong with them, it's that they just didn't react with anything. Maybe there's no space, or they were not being friends with other amino acids. What I mean by that is there's different R groups. Remember, there's charged R groups, uncharged, polar, non-polar. Right? Some things can interact with stuff and some things can't. So maybe these amino acids on the end couldn't react with anything. So it's basically a huge noodle soup is what we just created. And that's a tertiary structure. So just a bunch of alpha helices together in beta sheets. That's literally what level 3 is, tertiary structure. Now, level 4, or the quaternary structure, is when multiple subunits come together to create a protein. So let's back up for a second. This right here is not a protein. This is one subunit of a protein. Now, when we combine multiple subunits together, then we get a protein. So they start bonding with each other. So multiple subunits, a lot of alpha helices, a lot of beta sheets come together from different subunits, different polypeptide chains, and we get a protein structure. Now, this protein has 334 amino acids to it. It looks like nothing, but I drew this specific protein for a reason, and it's actually super interesting. Guess what this is? I bet you can't. This is what a potassium channel looks like. I know, right? You wouldn't think a potassium channel looks like. Because when we draw potassium channels, right, we draw them like, for example, here's like the lipid membrane, bilayer right here, and then we would draw like a potassium channel, right? And potassium would go through. Well, that's a simple hand way. The real way and how they actually look on a molecular level is this noodle-looking thing. This is a potassium channel. Isn't that super, super cool? I want to even blow your mind even more. This website is called AlphaFold. This right here is a protein. And you can literally expand, zoom in, do whatever you want. Look at it in a 3D way. You can even go to individual amino acids. So amino acid, for example, 93, right? Or let's say, actually, let's not do the end. Let's do something in the beta sheet. Okay, look, this is part of the beta sheet of the amino acid of the protein, proline. This is amino acid 11. So this section here is the amino acid. This is an L-type calcium channel in the atrium. I actually just made a video on the pacemaker action potentials. So that's why we did this. This is an L-type calcium channel in the atrium. This is part of the SA node. In this protein structure, this is actually from a dog. They don't have all of the structures from humans because it's kind of difficult to do it. So sometimes they have some from animals, like they have wolves, they have fish, they have dogs, they have anything. And the reason they're in different colors, as you can see, there's like different colors everywhere. The reason is because it's how sure they are that it looks like this. Right? Everything's a theory. So they hypothesize that maybe this section, the really dark blue section here, they're really sure that's what it looks like. But maybe some other parts, so let's say this yellow section here, they have a low confidence interval. Maybe they're not too sure that's what it looks like. They don't, it's a science, right? Everything's theory-based and hypothesis. Isn't that cool? So this website, let me just write it down here for you guys and experiment. It's called AlphaFold. One of my favorite websites to go to. They have so many proteins on there. You can look at what it looks, what they look like, how many amino acids are made of, what amino acids make up that protein. the alpha helices, the beta sheets, all that kind of stuff. Well, that's the big surprise. Hopefully you liked it. If you found this video helpful, please like and subscribe. And until next time, later.